So today I'll be looking at a few different types of mutations, and I have the original coding strand here. I've drawn out the complementary template strand. I've drawn out the mRNA that is transcribed from this DNA, and then I've also drawn out the amino acid sequence that is translated from the mRNA. First, I'll be looking at missense mutation, and this is where the amino acid ends up getting changed. And so I have the original coding strand here, and in this case, a substitution mutation would occur, which would change this G to a C. And that is a transversion mutation because we are going from a 2-ring purine base to a 1-ring pyrimidine base. And so I've illustrated the change in the DNA. We'd go from ATG to ATC, and this would result in an mRNA codon change. It would change from AUG to AUC. I've illustrated that here. This is the complete mRNA sequence, and this would be our new codon that would result from the mutation. And so as you can see, we would have 2 different amino acids produced, which would ultimately result in the function of the protein being affected. Next, we will look at silent mutations. And this is where the codon will change, but it will actually end up coding for the same amino acid. So I have the original coding strand here, and in this case, a substitution mutation has occurred where this C is substituted with a T. And this is a transition mutation because these are both 2-ring pyrimidine bases. And so here's the new DNA sequence. We have AAT instead of AAC. And this would result in the mRNA codon being changed to AAU, where it originally would have been AAC, but luckily these both code for the same amino acids, so the protein function would not be affected. I'll now be talking about a nonsense mutation, and so in the original coding strand, if we had an A inserted in between the G and the C, which would be a transversion, the new DNA sequence would now read TGA instead of TGC right here. And so our mRNA codon would go from a UGC to a UGA. And as you can see, this would go from originally producing an amino acid to now producing a stop codon, which would most likely result in a non-functional protein. Finally, we have a read-through mutation, and so here's the original coding strand, and if this A is substituted with a T, which would be a transversion, the sequence would now read TGT instead of TGA, and this would now result in an mRNA codon of UGU instead of UGA, and as you can see, this would go from a stop to coding for an amino acid, and this would just result in a non-functional protein as well.